So again, the gram-positive cell wall, very thick, about 20 to 80 nanometers thick, in fact, and it has the teichoic acid, which is involved in cell wall maintenance, kind of holding things together, enlargement during cell division, and acidic charge on the cell surface. So it gives the cell a negative kind of charge. The gram-negative cell has a thin layer, 1 to 3 nanometers thick, of peptidoglycan. It's somewhat rigid structure. The thinness, though, of the gram-negative gives it a flexibility. So a lot of times these are those spirochetes are going to be gram-negative because it gives them more like flexibility. And they are a little more sensitive to lysis by, changes in osmotic pressure. So if you have a very salty environment or a very watery environment, very hydrophobic or hydrophilic, it's going to be more sensitive to lysing for that reason. So non-typical cell walls, they do not have, well, they don't really have that structure of the gram-positive or gram-negative, but most of the ones that are non-typical are these Mycobacterium and Nocardia, which are called acid-fast. And we use an acid-fast stain to identify them. That's why they're called acid-fast bacteria. But they have this mycolic acid, which is a very long fatty acid chain, and it makes it very waxy, and it's resistant to chemicals and dyes. And so if you were able to get it to be dyed, if you're able to stain it, then it's going to be very difficult to remove the stain, and so it's called acid fast. So even acid alcohol cannot decolorize this, but acid alcohol can decolorize gram-positive bacteria as well as gram-negative. So mycoplasma is another one. Non-typical cell walls, they lack the cell wall structure of gram-positives or gram-negatives. They still have some peptidoglycan, but they also have this mycolic acid or cord factor, which is consisting of very long fatty acid chains, and it makes them more pathogenic. It's resistant to chemicals and dyes, and we use the acid-fast stain to diagnose it because they are resistant to colors, dyes, and if you do happen to get the dye on there, then they're going to resist decolorization, even with acid alcohol. We use this to determine or diagnose tuberculosis and leprosy, which are two of the Mycobacterium species. Nocardia is another bacteria group that is acid-fast, but this is probably the main one that we think of when we think of acid-fast bacteria. So the mycoplasmas naturally lack a cell wall. They do not have peptidoglycan, so they're very pleomorphic. Their size ranges are really small, from 0.1 to 0.5 micrometers, and they are filamentous to coccus or donut shaped. So the cytoplasmic or cell membrane is 5 to 10 nanometers. So that is actually bigger, thicker than peptidoglycan in gram-negative bacteria. And it's about 30 to 40% phospholipids, so it's not all phospholipids. In fact, it's not the majority of the membrane. It's the proteins make up about 60 to 70% of the cell membrane. Mycoplasmas contain a high amount of sterols. That's like cholesterol, but it's not really specifically cholesterol, but it's like cholesterol. And that helps to maintain the cell structure, gives it a little more stability, just like it does, the cholesterol does in animal cells. The archaea bacteria contain branched hydrocarbons rather than fatty acids, and so it's a little different cell membrane structure, different linkages as well. And the functions, they basically have all of the functions of membrane-bound organelles. So they do synthesis. They have photosynthesis occurs here in the cell membrane. And proteins, well, not protein synthesis, but ATP is made here as well. And so they regulate transport just like any other cell. They allow nutrients in and they get rid of waste products. but it's where we make ATP, it's where nutrients are processed. Sometimes there's some digestion going on there, some nutrients are built. Photosynthesis, cell ATP synthesis or cell respiration, all that goes on here in the cell membrane. So it has a much bigger function because there are no membranes in the cell. So it kind of takes on all of the membrane-bound organelle functions. So it's selectively permeable. Water can go through and uncharged small molecules can go through, but you need special carrier mechanisms for most molecules to get through the cell membrane, and secretion of discharge of metabolic waste and into the extracellular environment is another job of the cytoplasmic membrane. The outer membrane of gram-negatives, this is unique to gram-negatives. They have the lipopolysaccharides, which is a polysaccharide chain that functions as a cell marker and receptor, but it's also an endotoxin. It stimulates fever and shock symptoms in us. So whenever it comes into contact, like a gram-negative cell comes into contact, it can cause slight inflammation and fever. So lipoproteins anchor the outer membrane to the peptidoglycan, and so they're important to keep the cell together. And then porine proteins span the outer membrane and allow only relatively small molecules to penetrate, and the size can be altered to block entrance of harmful chemicals, so they can detect something that's harmful and can prevent it from coming in. And they act as a defense against certain antibiotics. So some bacteria are naturally resistant to antibiotics that affect the cell wall or that affect the protein synthesis because they can't get past the outer membrane. So the gram stain procedure involves 4 steps. The crystal violet makes both gram-positive and gram-negative purple, so you can see that's what they look like at that point. Gram's iodine will add to or combine with the crystal violet to make a crystal violet iodine complex, and that makes it bigger and it gets trapped in the cell wall where it doesn't have much of an effect because the cell wall is so thin in the gram negatives, but it almost looks a little darker when you get the iodine on there. It almost looks black because the molecule is the dye is combined with the iodine. The alcohol will decolorize the gram negative, so it's loose. It loses, its outer membrane weakens, and the wall loses dye, and it either dissolves or it shrinks or something, but it is losing. It decolorizes very easily, while the crystal violet iodine complex is remaining trapped in the thick peptidoglycan of gram positives. And then when you decolorize, you have to color the ones that you decolorize, otherwise you can't see them. And so we put a counter stain or secondary dye, which is safranin, on, and that will color the gram-negative bacteria pink or red, pinkish red color. And but it's mostly masked by the purple color of the gram positives, and the crystal violet iodine complex doesn't really allow much of that red color to show through, so it's not really seen. There's no red color there, it's just pretty much purple. So crystal violet, the primary dye, iodine is this mordant, which changes the shape of the dye and makes it larger and traps it in the cell wall. The alcohol is the decolorizer, and safranin is the secondary dye or the counter stain. So we'll be doing that in class. This will help you with, if you haven't done it yet, this will help you with the cards that you need to fill out, or the flashcards, to help prepare for the lab exam. So primary dye, mordant, decolorizer, secondary dye. And then you're supposed to color in the circles that are in the lab manual and write the color in. So that's basically what goes in the table in the pre-lab. So the differences in the cell envelope structure. The outer membrane in gram-negative bacteria It makes them more resistant to antimicrobial chemicals. It makes them more difficult to kill or inhibit than the gram-positive bacteria. So because of that outer membrane, they are more difficult to kill and usually then end up being more pathogenic. So infections with gram-positive bacteria are treated differently than infections with gram-negative. So you can't really use much of a cell wall inhibitor with gram-negative bacteria because the chemicals can't get to the cell wall. So how we treat gram-positive and gram-negative is different, and that is why we need to know if they're gram-positive or gram-negative, why that's important. So the cell envelope and disease. The cell envelope can interact with human tissues and contribute to disease. There are proteins in the outer cell wall of gram-positive bacteria that can be toxic. So those teichoic acid, lipoteichoic. The mycobacterium has that mycolic acid that is harmful to human cells, and it makes it resistant and difficult to kill. And the macromolecules in the cell wall are foreign, so it stimulates our immune system. So that's one way it interacts with the human tissue, and sometimes it causes disease and sometimes it's just stimulating the immune response, but that is what our body detects first, and so that's why it's really important for us to know about the cell wall and cell envelope. So the concept check questions for section 4.3, list and describe 3 differences between gram-positive and gram-negative bacteria. And for the test, you should be able to, if I give you a list of structures, tell me if it's a gram-positive or gram-negative or both or neither. So I'm tricky like that. Describe how the outer membrane of gram-negative bacteria contributes to disease symptoms. And then describe 3 functions, there are many functions, but 3 functions of the cytoplasmic or cell membrane.